Welcome back to botany class. Let's look a glance of today's learning objectives. Today you are going to learn few 3-mark questions. Micropropagation, artificial seed, advantages of artificial seed, virus-free plants, germ plasm conservation, cryopreservation, intellectual property right, patent, biosafety, bioethics, ethical and legal social implications. These are the topics we are going to learn today. Let's start one by one. Before learning this micropropagation, there are two definitions: somoclonal variation and gametoclonal variation. What's meant by somoclonal variation and gametoclonal variation? First question, the definition, what is define somoclonal variations. Somoclonal variations are variations in the somatic embryo, or it's called as embryoids. Variations in the plants which is regenerated in vitro. So any part of the plant which produces a somatic embryo. So somoclonal variations observed in the plant parts which is regenerated in vitro. Likewise, gametoclonal variations, variations of the plants which produced by gamete or gametophyte. So here gametes, there are male gamete, either male gamete or female gametes. Here gametoclonal variations observed in the gametes or gametophyte. Gametophyte, either gamete or gametophyte. So these two definitions first you learn. So today's first question, define somoclonal variation and second question, gametoclonal variations. Then let's start this 3-mark questions. Micropropagation. Micro, you know the word micro. What's the meaning micro? Very small. So micropropagation, propagation of plants selected by any part of the few part, micro part, any small part of the plant. It may be the root, stem or leaf or any other part. So propagation, micropropagation, which develop into an entire plant. An entire plant is in this micropropagation, number of plantlets will be produced within a single cell or single micro part. With a small part of the plant produces many plantlets. So it's commonly seen in pineapple, banana, strawberry and potato. These are the examples of micropropagation. The plants used to micropropagate is pineapple, banana, strawberry and potato. So this is another question, 3-mark question. Next question, artificial seed. This artificial seed is otherwise called synthetic seed. Synthetic seed, or it's called as syn seed. There are different names given for this artificial seed. What are the other names of artificial seed? Synthetic seed or syn seed. They are developed from the embryoid. So this embryoid is developed from somatic cells. Any part of the plant, it may be root, stem or leaf or any other part, it directly produces the embryoid. So this embryoid further it develops into a plantlet. So it allows to regenerate, it develops as a plantlet. So later it contains, first it forms a tissue, that is callus, that is a mass of tissue. From the mass of tissue it derives, the other parts are developed from this. So this artificial seeds, there are some advantages of artificial seeds. And this cell, mass of cell, this callus is the undifferentiated mass of tissue. From this, it develops the large nucleus and dense cytoplasm, and all the starch, oils and proteins, all the other parts derived from this callus, and it developed into the embryo, and this embryo is coated with agarose or sodium alginate. So protectants, to protect this, it's enclosed by, it's covered by this agarose or sodium alginate for preserve this seed. For a long time, this seed can be preserved, artificial seed. What are the advantages of artificial seed? This artificial seed can be produced, millions of seeds can be produced. In this method, the artificial seed or synthetic seed or syn seed or this artificial seed produce millions in number. And the seed, it's a easy method to produce because we do not wait for any season. At any season it can be produced. Easy method to produce and easy to test the genotype of the plantlet. The plantlet's genotype can be very easily tested by this method, and for long time it can be stored. The normal seed we cannot store for a long time. It can be stored very long period, and it produces identical plants. All these seeds, artificial seed or synthetic seeds, it produces the identical plants. Then the dormancy period can be reduced. A dormancy period is the resting period. In each and every species there is a dormancy period of the seed, but this seed, this artificial or synthetic seed, it reduces the period of dormancy. It breaks dormancy. This question, advantages, it may ask for 5 mark question, or the artificial seed, a short note, 3 mark question. Prepare it for 3 mark. Then virus free plants. Already this topic we learned previously. Virus free plants. What's this virus free plants? How it develop? The virus free plants, you know that so many pathogens like fungi, bacteria and viruses which affects the crop. The crop plants can be very easily affected by these pathogens. But the fungus and bacteria, fungi as well as bacteria, can be eradicated by chemical eradications. Chemical pesticides or fungicides or chemical substances can be eradicate these pathogens. But viruses, it cannot be eradicated very easily. So the virus free plants, transgenic plants can be developed. So it's possible to develop from the shoot apical meristem culture. Shoot apical meristem culture. So selected is shoot apex. Here shoot apex is selected, and from this region the tissues are derived and allow you to culture in the nutrient medium, and this shoot tip do not affect by virus. This is the reason, because of this reason the shoot apical meristem is selected for culturing virus free plants. Viruses which invade through roots, so when the plants are developed from the shoot apical meristem, the virus cannot enter into this plant. That's the reason it's selected as shoot apical meristem. The virus free plants are obtained by shoot apical meristem culture. This is one of the method to culture or to develop a plant. Then germ plasm conservation. Germ plasm. What's this germ plasm conservation? Germ plasm conservation is a method of conservation of living material, plant material like seed, pollen, etc. The seed or pollen or tissues or organ. These parts of the plant can be preserved in live form. So germ plasm conservation is a method of conservation. So this living material, seed, pollen and tissues are conserved conserved or protected, or it's conserved, it's stored. So this seed or pollen can be stored in the bank, seed bank or pollen bank. It can be stored to improve the plant breeding technique. In biotechnology, it's very much useful for producing improved varieties. Qualitatively as well as quantitatively, the crop can be improved by producing different new varieties. And this seed and pollen are conserved, stored in the form of pollen bank and seed bank. Likewise, the DNA, the gene bank or DNA bank, it can be stored. This preservation or the conservation, this plant materials are conserved for very long periods. So this can be used for researches, biological researches. This conservation technique is important by cryopreservation method. Next method is preservation method is cryopreservation. Cryopreservation, in the first chapter you learned this question, what's meant by cryopreservation? It's the process of preserve or conserve this plant materials like seed, pollen or tissue or any part of the plant can be preserved in very low temperature like minus 196 degrees Celsius. So minus 196 degrees Celsius in liquid nitrogen. It's stored in liquid nitrogen. It's the preservative agent, liquid nitrogen. The plant parts are preserved, mainly the pollen or the seed can be preserved, or tissues, organ or organelles, any part of the plant can be preserved extremely low temperature, that's minus 196 degree Celsius. So this, when it is preserved in very low temperature, it inactivates the activity of its enzyme and chemical substances. Chemical activities and enzyme activities will be stopped because of this very low temperature. So this, before preserving this, when it is frozen under very low temperature, it can be protected from stress. So it's coated with or it is immersed with protective agents. So before preserving, the plant material can be underwent protective agents. The protective agents are dimethyl sulfoxide, glycerol or sucrose. So these three are act as a protective agents. 2 more question or 3 more question, very important. What are the protective agents of cryopreservation? The protective agents of cryopreservation is dimethyl sulfoxide, glycerol or sucrose. Dimethyl sulfoxide, glycerol or sucrose are used as a protective agents. So after preserving, when it is used for research or when it is used again, it can be slowly transferred to room temperature. To make it active, the parts of the plants can be slowly transferred to the room temperature and it will be active. So this technique is called as cryopreservation. Important 3 more question. The next question, intellectual property right, which is commonly called as IPR. IPR in India, intellectual property right. In biotechnology, the microorganisms or the plants can be altered. Genetically, it can be altered. Manipulation of gene. So by genetic engineering technology, the microorganisms, natural microorganisms or natural plants can be altered into artificial. For this, each and every country gives the intellectual property right to the particular person. So here in India, this is IPR in India. So it's the right given to the particular person who who invented the particular product. So, it is the right given to the product. They have the right to sell and copyright and given license, that is patent. So, it includes patent, trademark, copyright, plant breeders right as well as trade secrets. So, everything is owner of the inventor or the discoverer. So, intellectual property right is given only to the discoverer or the inventor. It includes patent, trademark, copyright as well as trade secrets. So, patent, what is meant by patent? Patent is the authority given to the particular persons to give in the form of license, or a personal property. He has the license to sell the property. Others cannot copyright, and using, selling, everything, the ownership given to the particular inventor or the discoverer. So, patent in India, there are a few examples. You study basmati rice as well as some examples. So, in America 17 years of patent given, but in India only 5 years of patent can be given. So, it depends upon the country. Each and every country, the patent period can be altered. It depends upon the country. So, it is the intellectual right to the particular person of the others cannot copyright the same, or they cannot manufacture the product, or they cannot sell the product. They do not make, they cannot use this, they cannot sell it without the permission of the particular inventor or the discoverer. So, particular years the patent can be authorized to the owner or the discoverer. Then, biosafety. What is meant by biosafety? So, it is the method of safety, prevention of loss. Biosafety, there are so many products. So, in this chapter you learn in biotechnology, there are number of microorganisms can be altered through genetic engineering technology. So many byproducts are produced from the plant materials. So, here biosafety. So, in industry, commercially large scale production of these products, very easily it get spoiled, so many products. So, biosafety is prevention of this large scale loss. So, even the plants, it cannot protect it from the pathogens, easily it get affected and it affects the economic value. So, this is the method. Biosafety is the prevention measure given to the large scale loss. It avoids the loss of large scale. So, handling. So, lab, so many experiments which is tissue culture experiments can be done in the lab. So, lab facilities is very important. Otherwise, if it is not carrying proper, there are hazardous employing ongoing, and it is a risk for managing the hazards. There are a lot of hazards can be developed, and sometimes the protocols, if it is failure of protocols, the procedures can be followed when it is going on failure, it also causes exposure of biohazardous or pathogens. So, biosafety is very important when the product is manufactured in the lab. So, next one is bioethics. So, bioethics is it refers to ethical issues. So, ethical issues in the sense it is the moral issues. So, concerned with the ethical sometimes questions, ethical questions can be raised that arise from the relationship among life sciences, biotechnology and medicine. So, it is in the field of biotechnology, medicine as well as life sciences, the life sciences, biotechnology as well as medicines. Finally, there is a question, that is genetic engineering appraisal. So, this genetic engineering appraisal is an apex body under this environment, forest and climatic conditions. Environment, forest and climatic conditions, genetic engineering appraise. So, it related to hazardous microbes. So, regulating the manufacturing and use, the import and export, storage of hazardous microbes. It related to the storage of hazardous microbes. So, finally, in this chapter you are conclude this topic by future of biotechnology. What is the future of biotechnology? It will be future of biotechnology, future biotechnology. Future of biotechnology will be excited because you know there are number of experiments have been done within the laboratory. So, within a short period of time there are quality and quantities of products can be developed. So, in future, so it will be excited and it lead new science revolution. So, we can expect a science revolution in the future in biotechnology. So, this chapter, 5th chapter, we are going to conclude by this future of biotechnology. So, once again let us review what you learned today. First definition, somoclonal variation. The next definition, gametoclonal variation. Then 3 mark question, you prepare micropropagation. Artificial seed and advantages, prepare it for 5 mark. Then virus free plants, these all 3 mark question. Germplasm conservation, cryopreservation, sometimes it may ask for 5 mark question. Then this intellectual property right and patents and biosafety, bioethics. So, this IPR in India, which can be given property, it's the property given to the discoverer, full rights to given to the discoverer. It is protected by laws which is framed by the country. So, learn all these questions and you have the exam, half-yearly examination. Prepare first 5 lessons in botany. Thank you.